The Bcl-2 family of proteins are major regulators of programmed cell death, or apoptosis, in multicellular organisms. This presentation will explore groundbreaking research on the Bcl-2 proteins, examining how their molecular structure and function has been conserved from the earliest multicellular animals to mammals. We'll focus on the Bcl-2 proteins in Trichoplax adirans, one of the most primitive metazoans, to understand the ancient origins of this critical apoptosis control mechanism. The 3D molecular structure visualization on this slide highlights the vibrant colors and protein ribbons that characterize these important biological molecules. Apoptosis is a critical cellular suicide mechanism that shapes our bodies during development and maintains tissue homeostasis throughout life. Proteins in the Bcl-2 family are the key regulators of apoptosis, and were originally discovered in chromosomal translocations in follicular lymphomas. Bcl-2 homologs have now been found in almost all multicellular organisms, highlighting the fundamental importance of apoptosis in the evolution of multicellular life. This image shows a microscopic visualization of a cell undergoing apoptosis, with visible membrane blebbing and nuclear fragmentation, clear hallmarks of this process. The Bcl-2 protein family is central to the regulation of apoptosis, or programmed cell death. There are three main groups within this family: anti-apoptotic, pro-apoptotic multimotif, and BH3-only proteins. Anti-apoptotic proteins like Bcl-2, Bcl-XL, and MCL1 inhibit cell death and promote cell survival. Pro-apoptotic multimotif proteins like Bax and BAC trigger mitochondrial outer membrane permeabilization, which leads to cell death. BH3-only proteins like BID and BIM act as sensors and activators, tipping the balance towards cell death. All Bcl-2 family members share 1 to 4 conserved Bcl-2 homology, BH, motifs, as well as a hydrophobic C-terminal transmembrane domain. The overall balance between pro- and anti-apoptotic Bcl-2 proteins determines whether a cell lives or dies. The key difference in apoptosis control between roundworms and vertebrates is how the Bcl-2 family of proteins regulate the activation of caspases. In the roundworm C. elegans, the Bcl-2 protein SEED9 binds to the caspases, activating protein SEED4. Apoptosis occurs when the BH3-only protein EGL1 binds to SEED9, releasing SEED4 and activating the caspases SEED3. In vertebrates, the anti-apoptotic Bcl-2 proteins repress the pro-apoptotic Bax and BAC proteins. When this repression is relieved, Bax and BAC permeabilize the mitochondrial outer membrane, releasing cytochrome C and activating the caspases cascade. So the key difference is that in roundworms, Bcl-2 directly binds the caspases activating protein, while in vertebrates, Bcl-2 proteins control the mitochondrial pathway that leads to caspases activation. Trichoplax adirans is one of the simplest multicellular animals, with a flat disc-like body and only 6 cell types. As a placozoan, it represents one of the most basal branches of metazoans, providing insights into early animal evolution. Multicellular animals first appeared around 780 million years ago during the late Precambrian period. Studying the genome of T. adirans has revealed the presence of key regulators of apoptosis, like caspases and Bcl-2 family members. This genomic information helps us understand the origins and early evolution of fundamental animal processes. This slide provides an overview of our research identifying Bcl-2-like proteins in the Trichoplax organism. We started by doing an extensive genome screening of the Trichoplax adirans GREL BS 1999 strain, which identified 4 protein encoding sequences that potentially correspond to Bcl-2 homologs. To confirm the expression and sequence of these 4 TRBCL2-like genes, we performed reverse transcriptase PCR using specific primers. The confirmed sequences were then deposited into the NCBI databank under the accession numbers shown on the slide. This lays the groundwork for our further investigation into the function and significance of these Bcl-2-like proteins in Trichoplax. The slide presents the structural characteristics of 4 Bcl-2 proteins found in the organism Trichoplax. All 4 proteins exhibit the typical multimotif BH organization, with a transmembrane, trademark, motif at the C-terminus, and an all-alpha helical secondary structure. Analysis of the Bcl-2 protein highlighted a unique S-T-rich region between helices 1 and 2, which is predicted to be a mucin-like domain often found in secreted proteins. The 3D protein structure visualizations show the ribbon models of each protein, with the alpha helices colored differently. This information provides insights into the structural features of these Bcl-2 proteins, which are important for understanding their potential functions. Trichoplax is a simple marine animal that has a very basic body plan, yet it contains Bcl-2 proteins, which are important regulators of apoptosis or programmed cell death. We have identified 4 Bcl-2 proteins in Trichoplax: Bcl-2L1, Bcl-2L2, Bcl-2L3, Bcl, and Bcl-2L4, Bak. Bcl-2L1 and Bcl-2L2 cluster phylogenetically with anti-apoptotic Bcl-2 proteins, suggesting they may have a similar function in preventing cell death. In contrast, Bcl-2L3, Bak, and Bcl-2L4, Bak, cluster with pro-apoptotic Bak proteins, implying they may promote programmed cell death. The molecular weights and calculated isoelectric points, pi, for each of these Bcl-2 proteins are provided, which can give insights into their structure and potential interactions. The slide presents the genomic organization of 3 Bcl-2 genes in the organism Trichoplax. The first gene, Bcl-2L1, has a relatively long BH2 splitting intron of 1991 nucleotides. The second gene, Bcl-2L2, also has a long BH2 splitting intron, but slightly shorter at 1725 nucleotides. The third gene, Bcl-2L3, has a substantially shorter BH2 splitting intron of only 463 nucleotides. These differences in intron length between the 3 Bcl-2 genes suggest potential functional or regulatory differences between them. Understanding the genomic organization of these genes can provide insights into the evolution and diversity of the Bcl-2 gene family in Trichoplax. The image shows a fluorescence microscopy experiment visualizing the subcellular localization of Trichoplax Bcl-2 proteins tagged with a fluorescent marker. We performed these immunofluorescence experiments in mammalian cells transiently expressing the N-terminally flag-tagged Trichoplax Bcl-2-like proteins. This allowed us to observe where these proteins localize within the cell. To confirm the observations from the immunofluorescence, we also did subcellular fractionation experiments using baby mouse kidney (BMK) cells that lack the endogenous Bak and Bak proteins. This helped us isolate the different cellular compartments and determine the localization of the Trichoplax Bcl-2 proteins. Finally, we looked at the membrane insertion properties of the Trichoplax Bcl-2 proteins using purified yeast mitochondria treated with sodium carbonate. This allowed us to assess how strongly these proteins associate with membranes. Trichoplax is a simple marine animal that has been a focus of research on the evolution of cell death pathways. This slide examines the localization patterns of 3 Bcl-2 family proteins found in Trichoplax. Bcl-2L1 is detected in the mitochondrial and membranes, and is tightly inserted into the mitochondrial membrane. Bcl-2L2 is also found in mitochondrial and membranes, but causes the mitochondria to cluster around the nucleus. Some is also found in the cytosol. Bak and Bak are primarily localized to the mitochondria, with minor amounts in the microsomal fraction. The distinct localization patterns of these Bcl-2 family proteins suggest they may play different roles in regulating cell death and survival pathways in this early branching animal lineage. The C-terminal hydrophobic helix is critical for membrane localization of the Bcl-2 family proteins. Mutants lacking the transmembrane, trademark, motif display a cytosolic distribution instead of the normal mitochondrial localization. Proper subcellular localization to the mitochondrial outer membrane is essential for the apoptotic activity of these Bcl-2 family proteins. When expressed in COS7 cells, the truncated forms of Bcl-2L1, Bcl-2, and Bak that lack the trademark motif showed a mainly cytosolic distribution, highlighting the importance of the trademark motif for their localization. For the Bak protein, the 9th helix encompassing the trademark motif is particularly critical for its localization to the mitochondrial outer membrane. The slide presents data on the apoptotic activity of different Bcl-2 proteins from the organism Trichoplax. We can see that expression of TRBCL2L1 and TRBCL2L2 did not significantly increase caspase-3 activation in HeLa cells, indicating they are not pro-apoptotic. In contrast, expression of TRBAX and TRBAK led to substantial caspase-3 activation, similar to the zebrafish BAX ortholog. This suggests that TRBAK and TRBAX are bona fide pro-apoptotic Bcl-2 proteins in Trichoplax. The data highlights the different functions of these Bcl-2 family members in regulating apoptosis in this organism. The slide presents data on the anti-apoptotic activity of two Bcl-2 family proteins, TRBCL2L1 and TRBCL2L2. It shows that expression of these proteins led to a decreased percentage of caspase-3 positive cells compared to control cells when treated with apoptosis-inducing agents like thapsigargan, etoposide, and staurosporine. This demonstrates that TRBCL2L1 and TRBCL2L2 are indeed anti-apoptotic Bcl-2 family members. The bar chart provides a visual representation of the protective effects of these proteins against apoptosis induced by the different agents. The key takeaway is that TRBCL2L1 and TRBCL2L2 have the ability to inhibit apoptosis, which is an important function of Bcl-2 family proteins. Zebrafish embryos are a useful model for studying the activity of the Bcl-2 family of proteins, which regulate apoptosis or programmed cell death. Before the gastrulation stage, around 6 hours after fertilization, zebrafish embryos are naturally resistant to apoptotic stimuli. Expression of the pro-apoptotic proteins TRBAX and TRBAK was highly lethal, causing almost 100% embryo death by 6 hours post-fertilization. However, co-expression of the anti-apoptotic proteins TRBCL2L1 and TRBCL2L2 completely inhibited the pro-apoptotic effects of TRBAX and TRBAK. This demonstrates the protective role of the Bcl-2 family in regulating apoptosis during early zebrafish development. TRBAX is a protein that rapidly translocates to the mitochondria when expressed in cells. It forms oligomers in the mitochondrial outer membrane. This causes the mitochondrial network to fragment. TRBAX triggers the release of cytochrome C from the mitochondria. When expressed in BAX double knockout cells, most of the TRBAX expressing cells were positive for activated caspase-3, demonstrating that TRBAX is a bona fide BAX-like pro-apoptotic protein. TRBAK is a BH3-only protein that does not directly induce apoptosis, but rather ACTS as a sensitizer. On its own, TRBAK expression does not trigger cytochrome C release or significant caspase-3 activation. However, TRBAK can inhibit the anti-apoptotic activity of proteins like TRBCL2L1 and TRBCL2L2. This suggests TRBAK neutralizes anti-apoptotic Bcl-2 proteins, rather than directly activating the apoptosis pathway. So TRBAK functions as a sensitizer, making cells more susceptible to apoptosis by inhibiting the anti-apoptotic Bcl-2 proteins. The slide discusses protein-protein interactions between truncated, trail, Bcl-2 proteins, which are key regulators of apoptosis or programmed cell death. FRET, fluorescence resonance energy transfer, analysis was used to study the interactions between EGFP-tagged TRBAX, TRBAK and flag-tagged TRBCL2L1, TRBCL2L2. Co-immunoprecipitation experiments confirmed the interactions between the anti-apoptotic TRBCL2 proteins and the pro-apoptotic TRBAX and TRBAK. Interestingly, the interactions between the anti-apoptotic TRBCL2 proteins and TRBAX were disrupted by the presence of TRBAK, suggesting that TRBAK ACTS as a sensitizer BH3-only protein. The FRET efficiency between EGFP-TRBAK and flag-TRBCL2L1 was very close to the positive control, indicating a strong interaction, while the interaction between EGFP-TRBAX and flag-TRBCL2L2 was much weaker. These observations support the idea that TRBAK plays a crucial role in regulating apoptosis by disrupting the interactions between the anti-apoptotic Bcl-2 proteins and the pro-apoptotic Bcs. The table shows the binding affinities between different Bcl-2 family proteins, as measured by isothermal titration calorimetry, ITC. Bcl-2 binds to the BH3 domain of Bcs with a dissociation constant, Kd, of 426 nanometer, indicating a strong interaction. In contrast, Bcl-2 binds to the BH3 domain of TRBAK with a weaker affinity, with a Kd of 1.43 μm. Bcl-2 binds to both the TRBAK and Bcs BH3 domains with similar nanomolar affinities, with Kds of 563 nanometer and 728 nanometer respectively. These differences in binding affinities suggest that the Bcl-2 family members have distinct roles in regulating apoptosis pathways. The crystal structure reveals that the TRBCL2L2 protein adopts the classic Bcl-2 globular helical bundle fold, which is also seen in human and sponge Bcl-2 homologs. The TRBCL2L2 protein uses its canonical Bcl-2 ligand binding groove, formed by helices 2 to 5, to engage the BH3 motif of the TRBAK protein. Key interactions at the binding interface include the hallmark ionic interaction between TRBCL2L2R97 and TRBAK-D118, as well as additional ionic interactions and hydrogen bonds. The TRBAK residues I109, L113, L116, and Y120 protrude into 4 hydrophobic pockets on the surface of TRBCL2L2, further stabilizing the complex. Trichoplax is an ancient marine organism, the simplest known multicellular animal. The structure of its Bcl-2L2 protein has been refined to high resolution, showing clear electron density. This ancient Bcl-2L2 protein has a very similar 3D structure to the modern human Bcl-XL protein. A detailed comparison reveals that the key binding interactions are conserved between the ancient and modern versions of these proteins. This demonstrates remarkable structural conservation of this important protein family across hundreds of millions of years of evolution. The existence of a conserved ligand binding groove in a trichoplax Bcl-2 homolog suggests the possibility of conservation of interactions between trichoplax-encoded Bcl-2 proteins and their mammalian counterparts. Both the trichoplax Bcl-2L1 and Bcl-L2 proteins were found to bind to the BH3 peptide of the human Bax protein, as well as the BH3-only proteins Puma, BIM, and BID, with high affinity. Interestingly, all 5 human anti-apoptotic Bcl-2 proteins displayed nanomolar affinity for the BH3 peptide of the trichoplax Bac protein, with Bcl-2, Bcl-XL, and A1, BFL1 being the tightest binders. These reciprocal interactions between the trichoplax and human Bcl-2 proteins suggest an evolutionarily conserved mechanism of regulating apoptosis across these distant species. The slide presents binding affinities between trichoplax and human Bcl-2 proteins, which are key regulators of apoptosis or programmed cell death. It shows that both TRBCL2L1 and TRBCL2L2 from the simple marine animal trichoplax can bind to human Bcl-2 family proteins like Bax, Puma, and BIM with remarkably high affinities in the nanomolar range. This demonstrates the remarkable conservation of these critical protein-protein interactions across vast evolutionary distances, from simple marine organisms to complex humans. The high binding affinities suggest these interactions play an important role in regulating cell death pathways in both trichoplax and humans. This evolutionary conservation provides insights into the fundamental mechanisms of apoptosis and highlights the value of studying simple model organisms to understand core biological processes. The slide shows the binding affinities between the TRBAK-BH3 peptide and 4 human anti-apoptotic Bcl-2 proteins, HSA1, BFL1, HSBCL2, HSBCL-XL, and HSBCLW. The binding affinities are measured in nanomolar, nanometer, dissociation constant, Kd, values, with lower numbers indicating tighter binding. HSA1, BFL1, HSBCL2, and HSBCL-XL are the tightest binders, with Kd values of 13 nanometer, 45 nanometer, and 197 nanometer respectively. HSBcl-2 has a weaker binding affinity with a KD of 563 nanometer. This demonstrates the remarkable conservation of these molecular interactions across 780 million years of evolution, highlighting the importance of these Bcl-2 proteins in regulating apoptosis. The slide presents the binding affinities of various human Bcl-2 proteins to the truncated Bcl-3, Bcl-3 peptide, as measured by isothermal titration calorimetry, ITC, experiments. HSMCl1 and HSA1, BFL1 showed the highest binding affinity, with dissociation constants, Kd, of 67 nanometer and 135 nanometer respectively, indicating they are strong binders of Bcl-3. HSBcl-2 and HSBcl-XL displayed moderate binding, with Kd values of 767 nanometer and 877 nanometer respectively. HSBcl-2 had the weakest binding, with a Kd of 3060 nanometer, making it a weak binder of Bcl-3. Co-immunoprecipitation assays confirmed that Bcl binds more efficiently to HSMCl1 compared to HSBcl2 or HSBcl-XL, consistent with the measured binding affinities. The slide presents research on the functional interplay between trichoplax and human Bcl-2 proteins. Cell death assays in BMKDKO cells showed that expressing certain human Bcl-2 anti-apoptotic proteins, like MCL1, A1, BFL1, and Bcl-2, decreased the number of caspases 3 positive cells when co-expressed with Bcl. However, co-expressing Bcl-2 and Bcl-XL with Bcl had no effect on its pro-apoptotic activity. This functional interplay correlates with the binding affinities measured by ITC, suggesting the importance of specific interactions between these proteins. The key takeaway is that certain human Bcl-2 proteins can protect cells from apoptosis induced by Bcl, while others do not, highlighting the complex functional relationships between these proteins. The trichoplax BH3 peptides, Bcl and Bak, can bind selectively to different human Bcl-2 homologs. This selective binding suggests the potential to develop targeted therapies using these BH3 peptides. When expressed in HeLa cancer cells, the EGFP-tagged Bcl or TRBA-KBH3 peptides can sensitize the cells to etoposide chemotherapy. The Bcl-BH3 peptide specifically sensitizes cells that overexpress the human MCL1 protein. In contrast, the TRBA-KBH3 peptide sensitizes cells that overexpress the human Bcl-2 protein. These findings indicate that BH3 peptides from ancient Bcl-2 homologs like trichoplax could serve as templates for developing new BH3 mimetic drugs for targeted cancer treatment. The study of trichoplax Bcl-2 homologs reveals important evolutionary implications. We see a conserved core apoptotic machinery that has been preserved for over 780 million years, demonstrating the fundamental importance of this process. All Bcl-2 genes share a conserved BH2 splitting intron, suggesting they evolved from a common ancestral gene. The BH3-only proteins may have evolved from these more complex, multimotif Bcl-2 proteins over time. These findings highlight the deep evolutionary roots of the Bcl-2 protein family and the critical role it plays in regulating cell death across a wide range of multicellular organisms. The slide discusses the origin of BH3-only proteins, which are a type of Bcl-2 family protein involved in regulating apoptosis. It notes that no BH3-only proteins were detected in the genome of the simple animal trichoplax, suggesting the original Bcl-2 family may have consisted only of multimotif members. The slide then introduces TRBAK, a Bcl-2 protein in trichoplax that functions like a BH3-only sensitizer protein, binding to and repressing the anti-apoptotic TRBCl2 proteins. Based on these observations, the slide proposes an evolutionary hypothesis that some BH3-only proteins may have evolved from an ancestral Bak-like protein that lost its MOMP, mitochondrial outer membrane permeabilization, activity early in metazoan evolution. The 3 images on the slide provide visual support for these key points about the origin and evolution of BH3-only Bcl-2 proteins. The Bcl-2 family of proteins evolved the ability to control apoptosis, or programmed cell death, very early in the history of multicellular organisms, with key structural and functional features conserved from the simple organism Trichoplax all the way up to mammals. The specific ways in which Bcl-2 proteins interact with each other to regulate apoptosis have also been remarkably preserved over 780 million years of evolution, highlighting how fundamental and important this process is for the survival and function of complex organisms. Understanding the ancient origins and conserved mechanisms of Bcl-2 proteins provides valuable insights that could be leveraged to develop new cancer therapies. By studying and characterizing Bcl-2 homologs from evolutionary distant organisms, we may be able to identify new drug targets or design improved BH3 mimetic compounds to selectively kill cancer cells.